Welcome to Swastome, modern solutions for healthcare, built for India's tomorrow, shaping the future today.
It is my pleasure to introduce Dr. Priyanka Ghosh, a postdoctoral research associate at Northeastern University in Boston. Dr. Ghosh works in the field of developmental cognitive neuroscience, with a strong focus on electroencephalography, or EEG. Her research explores how large-scale brain networks emerge and reorganize across development, with particular emphasis on EEG microstates and brain network dynamics. She holds a PhD in cognitive and computational neuroscience and brings extensive expertise in designing EEG experiments, analyzing large-scale neural datasets, and developing open-source tools for EEG analysis. Her work bridges rigorous neuroscience methods with computational and open science approaches. We are delighted to have her with us today and look forward to learning from her research and insights. Please join me in welcoming Dr. Priyanka Ghosh.
I am Priyanka Ghosh. I'm a postdoctoral research associate at Northeastern University, Boston. It was already introduced at the beginning. The topic for today's presentation is EEG in healthcare, today and tomorrow. But before we start with that, okay, I need to move my slides. How does that happen now? Yeah, before we start with that, let me introduce you to the common functional brain imaging techniques in 2026. The one on the left-hand side is an MRI scanner, as many of you would recognize, which enables us to measure functional magnetic resonance imaging, which essentially measures the blood oxygen level dependent responses from the brain, which is an indirect measure of neural activity. The one in the center is MEG, or magnetoencephalography, which measures the magnetic field that is produced by the moving electrical currents from the brain. The one on the right is EEG, or electroencephalography, that actually lets you measure the electrical activity from the brain directly. All these three techniques are non-invasive in nature, but EEG is the most scalable, portable, and affordable of them all. Let's start with a brief history of EEG. Hans Berger, who was a German psychiatrist, discovered EEG while searching for a physical basis of mental activity. In 1929, Hans Berger recorded the first human EEG using a galvanometer-like instrument, as you can see here, that converted tiny brain voltages from the brain to mechanical deflections, which were recorded by an ink on moving paper. Berger's work proved that ongoing brain activity is electrical and observable at the scalp, laying the foundation for the modern EEG in neuroscience and clinical medicine. So from there, we are here today where you can wear an EEG cap and dance and swim and whatnot. But why should we care about EEG relative to other neuroimaging modalities is the question. So the temporal resolution of EEG is very good. It lets you measure electrical activity or the activity of your brain in the time scale of milliseconds, whereas fMRI will let you do that in the time scale of seconds. Most of the brain processes that happen in the time scale of milliseconds. So that way, EEG is temporally very precise and get the exact thing that's happening in the brain. Secondly, participants are free to move through a variety of natural environments, like swimming, as you can see in the picture, like they are dancing and whatnot. You cannot have that flexibility with MRI, right? Because you have to be there inside the scanner. And because it's like a magnetic scanner, the MRI, there are a lot of contraindications as well. Like you cannot have any metallic implant or cochlear implant or a pacemaker because, as you can imagine, the MRI has a huge magnet whose strength is like 3 Tesla to 7 Tesla, which is 100,000 times stronger than Earth's magnetic field. So you have to be restricted with MRI as well as MEG, but EEG gives you that flexibility to move around. So you can get brain readouts while doing a lot of other activities. Um, EEG can be used with a range of population, right from the neonatal stage to old age. The elderly people can have claustrophobia, and most of them have cochlear implants, so that makes them not an ideal candidate to get into an fMRI scanner. But with EEG, you have the flexibility to record the elderly, and with neonates and infants, you can imagine it's very difficult to get brain recordings from awake infants, because the only way possible to get them inside an MRI scanner would be to either sedate them or wait for them to fall asleep. MEG, on the other hand, is equally good temporal resolution, and it has an excellent spatial resolution as well. But then again, it's very constraining, and it's also very expensive. So EEG is the cheapest of them all, as I mentioned, but yet it gives you a lot of things. So it's always a trade-off between the spatial resolution, like if you're really interested in the what is happening kind of information, EEG is the best tool to go for. But if you're interested in questions like where is a particular thing happening in the brain, MRI would give you better spatial resolution. But with advances in technology today, with EEG systems with more number of sensors, better post-processing algorithms, and head anatomy mapping, the spatial resolution of EEG is going to improve tremendously in the near future, and it has already done a great job in the last 5 years or so. So, how do we measure EEG? So, we basically read electrical signals from the scalp via sensors or electrodes, which are embedded in a cap or net, something like this. But to connect the scalp, we need to connect the electrodes to the scalp via electricity conducting substances, which is either a gel or a saline. These gels are electricity conducting substances, so they really establishes the contact of the sensor with the scalp, so that we can capture as much of signal as possible from the scalp, right? And you can definitely see here that application of a gel into electrode caps or nets is a time consuming process, because you really have to go into individual sensors and do that, which is not possible when it comes to the developmental population and babies, because they are obviously fussy. So for babies, what we do is we have a net, we dip it into the saline, and then place it on the baby's head and trick them into that this is something like a swimming cap, so just have it on your head for a while. But saline solutions dry up quickly, so we cannot have that with adults. So gel is more suited for adults. A saline solution is more suited for the developmental population. Also, there is a lot of neurotechnology that is progressing towards inclusivity of people with whom we can, from whom we can record EEG, because a lot of population had very thick hair. They were not the ideal candidates for whom we could record EEG. For example, people with braided hair, like you can see, or tightly coiled hair. So the technology, EGI Maxton is doing a great job in designing EEG sensors that can very effectively capture signals from these kind of hair types. So EEG setup looks something like this. After you've placed the cap is connected to an amplifier to amplify the brain signals, which is connected to a recording computer or an acquisition system, on which we could see what are the brain signals being recorded from a person's brain. This is a participant on whom the EEG cap is placed, who would actually be viewing a presentation computer or a stimulus computer based on whatever task is being presented to that person. So why do we need that amplifier, which you saw was plugged into that EEG cap? It's because the EEG signals are really coming from the brain, right? We are essentially recording the post-synaptic potentials from pyramidal neurons of the cortex, which is there in the brain. And then there are these three layers of meninges, and then there is this bony skull. Then there is a scalp from which we are really recording. So these layers really dampen the signals. that are coming from the neurons, and what you're effectively recording is signals, a summation of signals from millions of neurons that are underneath this particular electrode. So because of that dampening, we really need to amplify the signals. So once we do that, the amplification is done and the acquisition system has acquired all the signals. What we see is something like this on the screen. These are basically recordings from a human. The x-axis represents the time. So as in real time, you can see what is the brain activity of a person, which is being, so we are measuring the voltage fluctuations, which is in microvolts, in the order of microvolts. These are essentially voltage differences from a reference channel, and each line represents activity from a particular electrode. So if it's, for example, a 64-channel electrode, there will be a naming convention, which is known as the 1020 International System. So for example, FZ here means an electrode that is placed in the frontal lobe on the Z line, that is center line. So will be an OZ electrode, which means that the electrode is placed on the occipital lobe on the Z line. And then there is like O1, O2, O3, or so there is a naming convention, like the left-hand side has odd numbers and the right-hand side is even numbers. So that's how the naming convention goes, but that's pretty standard. And we, in our EEG caps, try to cover the whole brain. There are standardized number of sensors, which all companies manufacture, like there are 32 channels, 64 channels, 128 channels, and goes up to 256 channels. So based on what kind of activity or what is the purpose of our brain recording, we can decide onto how many channels we want to go for. So the signals that you see, the EEG signals, they are basically a summation of multiple frequencies, which could be broken down into its individual components. These components are definitely constant sine waves from a number of frequencies, but they're often clustered into frequency bands, as you can see. For example, frequency that is above 30 hertz is gamma, then there's beta 12 to 25 hertz, then there is alpha from 8 to 12 hertz, then there is theta. Don't go by this, but theta is a bit lesser. It's around 4 to 6 hertz, and delta are the slowest waves. That's from around 1 to 4 hertz. Anything above, let's say 30-ish is gamma, and then anything about 45 is more like a high frequency oscillation. That is not really pertaining to the cognitive processes of the brain, but we can talk more about it later. So what information can we get from these signals when we look at it? Meaningful information. So this is the recording from a person when the person's eyes are open versus a person's eyes are closed. So this is the voltage scale. It's around 10 microvolt of activity. So if I want to see what are the changes in the brainwave oscillations when in these two conditions, if I just look at the activity readout, it looks like there are high amplitude oscillations in the eyes closed conditions versus eyes open condition, right? That's all the information that I can get from the EEG readout. And if I have all the time in the world, then I could also count that, okay, this is 1 second. How many cycles of oscillation are there? Okay, there are 10 oscillations, then that means it's alpha. But that's not practically possible to sit and count the number of cycles, right? So what we do for that is something known as Fourier transformation, which enables us to get the power spectra, which has constituents from all frequencies. So like in this figure, you can see that most of the activity that happened was at 10 hertz, which is the alpha frequency. And also in the eyes closed condition, the power of alpha was, EEG power of alpha was higher than it was in eyes open condition. So, I mean, we could also see the EEG activity and tell that the amplitude of eyes closed condition was higher, but it was not meaningful enough because we didn't really know at what frequency was that happening. So power spectrum analysis lets us do that. And why did we select these 3 electrodes in particular, OZ, O1, and O2? So this is OZ, like occipital electrode on the Z line, and the neighboring ones are O1 and O2. The occipital lobe, as some of us would know, has a visual cortex. So changes related to eyes open and eyes closed would be more observable in the visual cortex, and that's why we went for these three electrodes. But do we always need to convert our signals into a power spectrum to get meaningful information? No, that is not true. You can also get a lot of information from just your EEG signals as is, of course, after removing all the noise and pre-processing the data. So something that you see here is called a pattern reversal checkerboard. It elicits a response in your brain known as a visual evoked potential. So whenever there is an event, a visual event, auditory event, or a tactile event, brain gives a response. Okay, so that response comes up as a particular wave pattern. The visual evoked potential in this case is a wave that is characterized by a P100, which means a positive peak that occurs at a latency of 100 milliseconds, right? So this is a very reliable marker in infancy to as a test for visual integrity. If there is an absent P100 in response to this checkerboard, or like a delayed P100, like it didn't occur at 100, it occurred at 150 or something, then one could say that there is some dysfunction in the visual pathway. So it's a reliable marker, and there definitely, this is a sensory level processing and can be tested for within the first year of life. So that's really amazing. Okay. So that is sensory level processing, and now we would see how EEG is helpful in higher order cognitive level processing. So for example, in this task where, so by a higher order cognitive processing, I mean cognitive functions like attention, working memory, decision making, etc. So like in this task, the participant was asked to report the net direction of the moving dot. So like you see in trial 1, these dots effectively move in a particular direction, which could either be left, right, top, or bottom. So the participant had to figure out that direction and press the arrow key in whichever direction the participant felt the dots were moving in. So that is the task, but to test it out for cognitive functions, there were some trials where a distractor dot was introduced, which was moving in any random direction, but the task was essentially the same. The participants still had to figure out the direction of the white dots. So because the distractor was introduced in a few trials, as you can see, this is ST, this is saliency trials. So you would remember the name. So the reaction times of trials in ST was higher than the reaction times of trial in WT, showing that a distraction was indeed into play when the participants were looking at these trials. And to have another sort of control trials, we had moving dots which were not really moving in any direction, but the participant was unaware of it. So essentially, there was no real response to this, but the participants still had to hit the response button. So whatever they thought was the best direction, they responded to it. But since they were figuring out for a longer time, you can see the reaction times for NT trials was the highest. So that means the participants were really engrossed into the task, and the reaction times do show that the distraction and the task difficulty that was introduced with no particular direction was affecting their responses or performance. Now we wanted to see, because behavior is just a manifestation of brain activity in a way. So what was really happening in the brain that gave rise to these longer reaction times, right? So if you see this power spectrum again, it's Fourier transformed EEG signals. The power spectrum corresponding to the trials with the distractor had a higher, significantly higher power between 8 to 9 hertz. So which means that there is some distractor suppression going on that is shown by these enhanced 8 to 9 hertz frequency, and that is really not happening for the trials of the NT condition or the control condition, even though it was a difficult task category. So the distractor suppression is really happening at that particular frequency. So you can get more out of this, what's happening in the brain. Like if I want to know, okay, this enhanced power is happening is at this frequency, but what are the regions which are really into play while there is an enhancement of this frequency band? So it's not the entire brain that is into distractor suppression, right? Because you have a task to complete, and there are other things also the brain has to process while doing the task altogether. So there are source localization techniques. There are a lot of inverse solutions out there in the market that lets you enable localize the sources of any brain activity fluctuation. And just not that, suppose you have localized the sources and then you have three nodes A, B, and C. You want to know that what is the directionality of the communication. Is there a causal node and is there a driver node? Like um, is the activity of one region signaling the activity of another region? They are communicating in some way or not? This is possible to measure by a technique known as dynamic functional connectivity. We, in our paper, had used range of causality for this metric. So what this essentially does is, if we reconstruct the time series at these localized regions, and suppose I'm able to predict the time series or the future events of time series Y by the past events of time series X, then that means that A is causing B somehow. And that is the basic idea behind looking at the directional connectivity between any two nodes. And because this is EEG and we have the time signals in real time, we are able to do this, which wouldn't have been possible with fMRI because it's in the order of seconds and these processes are happening in the order of milliseconds, right? So we're very close to getting the voxel level activation that we get through fMRI, but in a temporally precise manner. So there is another metric called EEG microstates, which I used in a recent study, which is a readout of large-scale brain networks based on the particular topographies that we get. As you can see here, these individual topographies are short-lived state that repeat over time, and they are representative of fMRI large-scale brain networks, which is very useful in looking at developmental trajectories. So in our study, what we did was we had longitudinal data, EEG data from 4 time points, as around 3 months, 6 months, 12 months, and 24 months from 557 infants. And what we essentially saw is when we extract the microstate durations of each of these recordings, we found that these durations decreased over time from 3 to 30 months, which was indicating rapid switching of brain networks, which is also supported by a lot of structural imaging studies, which show that as the infant matures, especially in the first 2 years where there's rapid maturation happening, there's increased myelination, which leads to rapid neural impulse transduction. So that faster conduction is being reflected here. So this is one of the major findings that came out of our study, and this is very helpful in conditions like ASD, because altered microstate dynamics have shown to be predictive of ASD much before the behavioral manifestation occurs, which is not before 3 years, right? So it definitely helps us in predicting ASD in the first 2 years of life itself. And this could really help us in designing better intervention strategies even before the behavioral manifestations come up. And just not in the developmental population, there are certain metrics that could be used for diagnosing Alzheimer's at an early stage. There's something known as phase lag index, which, as the name suggests, it measures the lag in phase of 2 time series. So I have region A, I have region B, there are 2 time series from those regions. And if the 2 time series show a consistently lagged phase throughout the entire recording, that means the two regions are synchronized. if their phase lag is changing over time when they are not synchronized. So, studies showed that a decrease in theta band temporal phase lag index is indicative of memory network breakdown in the elderly. This is very helpful for conditions of MCI and Alzheimer's because uh, a lot of people really diagnose these conditions when there is already a severe loss of network breakdown, or it's like it's at a very advanced stage. With a lot of EEG metrics, one could really uh, predict the onset of early Alzheimer's. So it is really helpful for diagnosing a lot of diseases from right from infancy to the elderly. EEG is also the gold standard for epilepsy. So uh, this is, I think, one of the first applications of EEG that has always been there. And epilepsy is characterized by seizures which have high frequency oscillations, as you can see here. Um, by looking at EEG patterns, one could say if the EEG is focal or generalized in nature, and also by looking at some connectivity dynamics, one could say if the epilepsy is drug resistant or drug responsive. So that could inform the treatment plan. EEG can also distinguish between the different stages of sleep, like there's stage 1, stage 2, stage 3, and REM. And every stage has a characteristic brain rhythm. So EEG is very helpful in diagnosis of sleep disorders like insomnia, narcolepsy. So there are some recent big advancements, like in the last 5 years or so in the field of EEG. So one of them is brain-computer interface that really promotes accessibility and inclusivity for disabled individuals by letting the brain connect with an external device. So what happens is, uh, since we can't measure the rapid brain signals, it can be decoded in real time, and then the commands could be used to control a cursor, a wheelchair, or any prosthetic device. So that is a huge application in BCI. Another application is at home, where you can do real world monitoring by wearing wireless wearables, EEG devices, and that lets you monitor your stress levels and mindfulness through neurofeedback, right? There, again, then there are a lot of cognitive training apps in the market, which some of you may be knowing about. All of them work with EEG, where you could really create a closed loop between the brain state and the behavior, and that can help you adapt to the game challenges level wise, and that will really help you control your attention and cognitive control more effectively. So all in all, as you could see, EEG is widely used in healthcare, and all because your brain is electric, right? So we are all electric, and we all have that spark in us, quite literally. So I will let you sit with that. Thank you so much.
Thank you so much, Dr. Ghosh, for such an insightful and engaging talk. The way you actually connected the fundamental research with real world impact, it was truly thought provoking and inspiring for our audience. I think we already have a question in the chat box.
Okay.
How does task based EEG versus resting state EEG differ in sensitivity and specificity for early Alzheimer's disease detection?
Uh, Honestly, I am not very sure of this answer because I do not have experience with Alzheimer's patients. But as far as the specificity is concerned, I think the way we go about any control, that is healthy control and the disease condition, is that if there is a task, we have certain brain readouts, be it in the power spectrum domain, phase lag index, or connectivity measures. We have it from the healthy control, right? So we know that this is how the healthy patterns look like. And then we have it from the Alzheimer's patient. And we really do not know what metrics would actually show differences. So some of it is definitely exploratory as to what metrics is diverging from what we got from the healthy controls. So that is about the specificity part of it. And I'm not sure if Alzheimer's patient will not be sensitive in picking up in any sort of signals, like especially in today's age when the post-processing algorithms have developed tremendously. Because I think the machine learns, and with a lot of learning, I think that sensitivity that can be taken care of. And so yeah, this is how I would go about it. Okay, I would move on to the next question. So are portable or low-cost EEG devices reliable for identifying EEG slowing in Alzheimer's disease compared with clinical EEG systems? So the portable EEG systems are very new in the market, and there are a lot of startup companies working on this, trying to get there to get the brain signals with as much precision as we get it through clinical EEG systems. But at this point, in today's date, it's still a trade-off between the precision and the portability, I would say. But this field is rapidly evolving. So we might not be very far from getting to a stage where we have a portable as well as precise enough EEG instrument to measure Alzheimer's and whatnot, right? So there's another question. As in fMRI, the spatial resolution is very high and the signal is very strong, but in EEG, the temporal resolution is high. So how can you extract the strong signal? Yeah, so the temporal resolution high. So that is why we have amplifiers which are connected to the head cap. So there are a number of ways that we ensure that we are capturing as much signal as possible from the scalp. So before we actually start recording from a participant, while we are gelling a participant or have the saline solution, there is something known as impedance, which is an obstruction to the electrical flow. So we have a screen where we can see what the impedance of each sensor, and we make sure that the impedance of each sensor is below 50 kilo ohms in order to capture the best signal quality as possible. So there are ways where we can ensure that, and then once the EEG data is collected, there is a rigorous pre-processing pipeline that the data has to go through in order to make sure that we are maximizing the signal to noise ratio in a way. So yeah, it goes through a lot. There are a lot of checks at different points, and yeah, that is about it. And the next question is, it was short and informative session, ma'am. So according to you, which types of EEG markers you found to be the most reliable across individuals and which are still difficult to translate? I mean, if an EEG marker is reliable, people would put in the effort to translate in some capacity or the other, right? But one of the most widely used markers historically is definitely the visual evoked potential marker that I showed you. There is another marker called the P300 marker, which, as the name suggests, is a positive peak at 300 milliseconds latency. It's an oddball marker. In fact, people say that is a marker that is used in place of the previous lie detection machines that you used to do. Because the oddball, that marker is about, that if there is a set of standards that you are expecting, let's say a blue square. You see a blue square, blue square, blue square, blue square, and suddenly there is a red triangle. That is an oddball, right? And when you see that, the brain elicits P300. So that is a better measure, which is used in place of the old traditional lie detectors, because people through yoga and mindfulness practices and all kind of could regulate their physiological functions a lot. So that is why a cognitive marker is more reliable in that sense. And there indeed are a lot of markers like alpha in the frequency domain is a very reliable marker. Like if you open your eyes, the alpha always decreases as compared to in resting state. And like I am sure a lot of you listen to these YouTube audios where they are like focus enhancing audios, like you keep it in the background, or music for work. What do they essentially tap into is these frequencies at which your attention is the best. So like beta frequency they would tap into, so that your brain also entrains into those frequencies, and then you can work better or you can focus better. So I think there are a lot of markers that are being used and translated as well. But yet there is a lot of potential to it. So that was a long answer to a short question, I guess. So thanks for a quick overview. I am currently a cognitive science student at IIT Delhi. I would like to know if you take interns in your lab. Okay, I can ask my supervisor and let you know for sure, Akshay. And based on your experience, how cautiously should researchers interpret power changes in specific frequency bands? Very cautiously. But I think if you're straight away seeing power changes in a frequency band, let's say in task A versus task B, uh, that is definitely a finding. That is the data speaking for you. But very recently, they are also kind of classifying the power spectra in terms of aperiodic component versus periodic component. For the very longest time, people only used to focus about the periodic component. But quite recently, we have found that the aperiodic component also holds a lot of information, which is nothing but the 1 by f scaling that we have. Like the lower frequencies always have a higher power and the higher frequencies have a lower power. So initially people would always take out the aperiodic component and literally throw it. But now people are really separating the aperiodic component from the periodic component and seeing if the differences between the two tasks is in this component or that. And obviously, I mean, there needs to be like you have it for any other experiment. Like there has to be a lot of other measures, like you see what is the power of the statistics and things like that to really make a claim of that sort. So that is that. There's so much that you can do, right? So I think that is the end of all questions. Any other questions?